All right, let's get going. Thank you guys for the interesting conversation in the chat. And Mark, I actually would agree with you. I think anthropomorphizing is something that we definitely need to always be thinking about and trying to limit. Right? Anthropomorphizing, for those of you that may not be familiar with the concept, is this idea that we sort of interpret things, especially when we're discussing other species um, on the planet. Although we do it also, in sci-fi, right? When we sort of have these fictionalized alien species, but nonetheless, anthropomorphizing, means that we're basically giving or imparting on actions and of other species, the reasoning that we would ascribe if another human did it, right? So we need to always be aware of this. Other species are not humans. We are very, very different in many, many ways, and the reasons that we do things are not always clear, even if it would be clear in another human. So that's definitely something I think that everybody should always be thinking about when working in biology. Okay, so welcome everyone. Let's get going. Doo, doo, doo. Okay, let's see. There we go. You should all see my screen now. Okay. All right. Our regular housekeeping slide. Today we're tackling chapter 28, which is on fungi. Fungi or fungi. For whatever reason, I tend to kind of go through across both of those pronunciations. Both of those pronunciations are completely correct. So don't ever stress about whether or not you are pronouncing it correctly. I'm sure there. So fungi, yeah, fungi is another way. There's going to be a lot of, you know, the entire field of fungus. Um, is filled with some really interesting vocabulary. So I'm sure especially with my Brooklyn accent, I'm sure we're going to mangle some mycology pronunciations today. So I apologize in advance. Okay, so you should have completed your pre-lecture assignment last night by midnight. Tuesday, you will be tackling 29, chapter 29, and I say you because today is the last day that I am lecturing for you. You will have Dr. Arnott. His first lecture will be on Tuesday. I spoke with him last night, and he was making some adjustments to what he's going to do as far as assignments. And so he asked me to let you all know that he will email regarding the assignments in the coming couple of days. So please, everyone, be aware and be on the lookout for sort of an introductory email with some instructions from Dr. Arnott. Okay, so now that being said, I will tell you that I have enjoyed lecturing in the course. I'm gonna miss being here with you guys, but that's okay. Perhaps you've had enough of me. And Dr. Arnott is actually really great and a true gentleman. So I think you guys will do well continuing with him. Okay, now regarding office hours, right? I'm not going to be lecturing regularly, so I'm not going to have the regularly scheduled office hours. But that absolutely, completely does not mean that I am not available. I am absolutely available to help with anything in the chapters that I have covered with you. All you have to do is email me, and we will work out a time. We'll find a time to meet, and then we'll create a Zoom link or something like that, and and then we can meet and discuss whatever questions or topics it is that you may want to discuss. Several of you have already done this across the first half of this semester, so hopefully it has been relatively easy and painless, and hopefully it remains that easy for everyone else. Okay. That being said, please remember that there are 4 chapters, including today's work, that we are covering together that will be covered on your second midterm. Okay, so your second midterm will be a combination of work that you've done with me and work that you will do with Dr. Arnott. Okay, that being said, last line here, Dr. Arnott's taking over lecturing duties on Tuesday. Okay. Any questions on any of the housekeeping before we get going? Okay,
Can you hear me,
for
Professor?
dates on midterms and exams, please consult your syllabus. Okay, yes, I'm sorry, go ahead. Who's speaking? I'm sorry.
Hi, Professor. Just a quick question. I'm not sure if it
Yeah.
might be different once Dr. Arnott takes over, but on our Achieve, so is the grade book on Achieve our collective grade book, or is it only our Achieve grade and then, exams and other stuff will be separate, or everything's just going to be on Achieve?
Right. So midterm 1 is in your Achieve grade book, but what is important for everyone to keep in mind with the Achieve grade book is that, Achieve is basically uninformed, right? So it's just scoring the things that we tell it to score, okay? Or maybe I should more properly say I tell it to score. It's not weighted. So in other words, it takes everything, to be of the same weight. And that's obviously not the case. This is all spelled out in your syllabus, and we talked about it in the first lecture. The exams are worth, a certain percentage of your overall course grade. Your homeworks, your iClickers, your participation, all these other things go into a separate part of your lecture component. Your labs are not on Achieve in the Achieve grade book at all, at least not that I am aware of. So just realize that what you're seeing in Achieve are your raw scores, okay, not your weighted scores. So it's good that you can kind of see how you're doing on each individual assignment or on your homeworks overall or on the exam, but remember it has to be it then has to be weighted. Is that helpful?
Yes, it is. Thank you.
Okay. Yeah, no problem. Okay, great. Any other questions before we move on? All right, let's get going. Okay. All right. Evolution and diversity in fungi. Our 4 key concepts for today. So we have kind of talked about fungi, sort of maybe just touching upon the topic in several different ways. So we're going to talk about really probably the most distinguishing characteristic of all fungi. Then we're going to talk about what roles they play in ecosystems. We'll of course talk about reproduction, and we'll finish up on some of the ways that we humans have used fungi for very long periods of time for practical purposes, some of them going way, way back, and the impact that it's had, okay? So the distinguishing characteristic of all fungi is that basically they digest food outside of their bodies, okay? This is called absorptive heterotrophy. Basically, they secrete enzymes, digestive enzymes that are going to break down their food, and then they basically bring in those digested molecules, those broken down, simplified molecules. This is really important because fungi are incredibly important for the whole biological ecosystem. But what's really different about this approach is that they don't have to ingest their food and then process it. right? So think about it. You can't eat. Anybody ever try and eat one of those like giant sandwiches, right? And you can't even like fit it into your mouth. Or what is that show on with Food Network or whatever one of them is, where it's like diners, drive-ins, and dives, right? And you have the guy who goes around the country to all these different places and tries their food. Yeah, Guy Fieri or something like that. And he'll have these giant burgers or giant sandwiches or what have you, and he's right, that kind of a thing. We have a limit. We have a limit to the size of food that we can ingest. Um, that's not the case for fungi. That you can think about if anybody's ever seen a snake eating, right? So snakes don't chew, they swallow whole. Birds also often swallow things whole. So there's very definitely a limit to what you can have. And snakes have this whole anatomy built around being able to ingest larger food items. Yeah, snake eating a rabbit. Right? I mean, it's amazing some of the things that they can do. But their head anatomy has specifically evolved for that purpose. The jaw is how it connects to the rest of the skull. Very, very different than how it is in mammals. Fungi don't have this problem. They don't have this problem at all, right? I mean, they could live on their food. They often do, right? They live on their food source, and they're just secreting out these digestive enzymes, break it down, and then take in those broken down molecules, at different levels of being digested. Okay. Many fungi are saprobes, okay, which means that basically they're going to absorb nutrients from dead organic matter, right? So something that's not living, okay? Some of them are predatory, some species are parasitic. And then amazingly, some of them are also mutualists. Some of them live in a cooperative relationship with another species. And this is incredibly important. And I hope I will be able to successfully stress how important some of these mutualistic relationships are. So let's sort of take a look at the phylogenetic tree for fungi. Best information right now tells us that fungi evolved from a protist that had a flagellum. The common ancestor of fungi in animals was probably similar to what are today called choanoflagellates. Choanoflagellates, the fungi and animals, actually form sort of a subclade that are called opisthecons. I told you we were going to have some interesting vocabulary today. Okay, so just to kind of get, and that's what you're seeing here, right? The opisthecons. And you can see, the fungi being this one branch, and that absorptive heterotrophy evolved on this branch. So all the fungi are going to have this absorptive heterotrophy, right, where they can digest their food outside of their bodies. So what makes fungi, right? What they're all going to have in common, they're definitely all going to have this absorptive heterotrophy in common, okay? Distinguishing characteristic of fungi. The second thing is chitin. Okay, chitin is a polysaccharide that is heavy on the nitrogen, contains a good amount of nitrogen. And this is what goes into making up those fungal cell walls. Okay, so this is another sort of characteristic. There are two main divisions when we talk about fungi. The unicellular, which for the most part, when we're talking about unicellular, single-celled fungi, we're talking about yeast. And then, of course, if you have unicellular, the other side of that coin is going to be multicellular. So let's talk a little bit about yeast. Here's a photo of Saccharomyces cerevisiae. This is the sort of prototypical yeast that is used in the lab for evolution experiments and all kinds of experiments. Yeasts are unicellular, they're free living fungi. It's not its own taxa, right? So this is not a monophyletic group. Yeast are sprinkled across the tree of all fungi. But yeast is more explains here. I use the word lifestyle, but I don't love that word, right? A way of surviving in the world. Okay, it has evolved several times within fungi. Um, Typically they need to live in very moist environments, so liquid or high humidity kinds of environments. They grow very easily when the conditions are correct or right for them. And they are incredibly important, yeast, for a lot of human uses. And this yeast are probably the sort of defined relationship that we've had for probably the longest period of time, of all the fungi, right? So we use them for baking. We use them for brewing. We use them for alcohol production. And what's interesting, and one of the things that I'd like to point out, is the oldest ways that we specifically use yeast, even before we knew we were using yeast, uh, have been about food preservation. Okay? Think about it. Food preservation. Baking is a way to preserve and use the grain that you have. Brewing is a way to preserve for some prolonged period of time the components that go into beer making, right? Which includes a grain and some other biological products, which would otherwise rot. Alcohol production also, and there are many, you make alcohol with many different kinds of grains and agricultural products. But nonetheless, doing that preserves it. And very interestingly, think about the time before modern medicine. Okay, think about the time before good sanitation. Think about the time before germ theory, when we didn't really understand what could make you sick, that germs were the cause of infectious disease. And drinking alcohol was actually something that was safe to drink. In many parts of the world, many different periods of time, it was much, much safer for your health to drink something with alcohol in it than it was to drink the local water. So think about the role that something like yeast has played in human health even before we understood it. Now, I want to call your attention to this photo. Here's a photo of yeast. You can see the individual cells. Here you see one yeast cell that's actually budding. This is asexual reproduction in yeast. They grow a bud, and then that bud, when it is ready, will detach from the mother cell, leaving all of these that are highlighted in yellow, all of these what are called bud scars. So you can actually tell by counting bud scars how many buds, how many asexual offspring a specific yeast cell has had. Okay. Let's talk about the multicellular fungi. Okay. When we talk about multicellular fungi, the body of the fungus is called the mycelium. It's typically composed of these tubular filaments called hyphae. And you can see a photo of hyphae and sort of broken down a little bit, a diagram of hyphae. Hyphae have cell walls that have that chitin in it. Okay. And some hyphae have incomplete cross walls. In other words, they're not completely walled off. That chitinous cell wall does not completely wall off one cell from another. Okay. Um, and when they hyphae without these septa are called coincidic. Okay, just to give you some vocabulary there. All right, so let's talk about the hyphae. Hyphae grow quickly. This is another really interesting component, aspect, phenotype of fungus, fungi. The hyphae can grow very, very quickly. In fact, the record for mycelium growth was over a kilometer long in a 24-hour period. So when I say quickly, I mean pretty quickly. Okay. Um, Hyphae can be reorganized into what's called a fruiting body. Mushrooms, as we see them and think about them, are actually fruiting bodies. Fruiting bodies are for spore dispersal. So fruiting bodies are for aiding in reproduction. Okay. Mycelial mass can be very large. In fact, there is a fungus in Oregon. It's mostly underground that covers over 900 hectares, which is more than 2,200 acres. Okay, acres. And the estimated mass is greater than even a blue whale, which is not only the largest animal currently existing, but to the best of our knowledge, the largest animal that has ever existed on the planet. Okay. Some fungi have these rhizoids, which are modified hyphae, specialized hyphae that anchor them to something, whether it is their food source or some solid substrate. All depends, but that's called a rhizoid. Okay. All right. So ecological ranges. This is another amazing uh, aspect of fungi, right? Um, so first of all, all right, let's try and tackle the slide as I put it together. Fungal mycelium has very large surface area to volume ratios. Think about it; they're very thin and very long. Lots of surface area, not so much volume. Okay, this is extremely good, beneficial if you take in your food by absorptive heterotrophy, right? You've got a large surface area. Surface area is directly related to how quickly you can bring things in. Okay. And this is one of the reasons why hyphae can grow so quickly. The longer hyphae grows, remember it's sort of tubular, right? The longer it grows, the more surface area it has without greatly expanding the volume. And you're bringing in more nutrients. You can put those all toward growth. Okay. Um, Water loss, by the way, is also high. That's the other side of that coin with high surface area. Really good at bringing things in, but you are susceptible to losing water. You've got a small surface area. You can't store a large volume of water, and it is all exposed pretty much in terms of surface area. So you can lose water quickly, which is one of the reasons why fungi prefer, do much better in humid or wet environments. Okay. Now, interestingly, they can tolerate hypertonic environments. What do we mean by hypertonic? Okay, that means there's a lot of dissolved particles in the, let's say it's in water, right? And the water outside of the fungus has a lot more dissolved particles than inside. That's okay for fungus. That's fine. This is the reason why you can get molds that will grow on food, typically rich food, in even the refrigerator. Okay, and the other reason that you'll get fungus that'll grow on a food, usually mold, right? In something like a refrigerator is because fungi can exist, can live, can survive in an extremely wide range of temperatures. -6 degrees Celsius all the way up to 50 degrees Celsius. And that, and we're not even really talking about at the very, very extremes. We're talking about, they can comfortably grow in that environment. Okay, saprobic fungi. So fungi are very important for Earth's ecosystems. I mentioned that a couple of times. Saprobic fungi, they are doing a lot of decomposing. They're breaking down dead organic matter. Okay, things that have died. They are kickstarting the decay process, and that decay process is key to the recycling of compounds and elements in the earth. Uh-oh, looks like something's up. Okay, do you have me now? Do you guys have me now?
No, it's just black.
Just black.
We hear you, but we don't see the slides. Yeah.
Okay, so let me stop the share and see what we can do here. Okay, have you picked up my camera?
Yes.
Okay, so you can see me. All right, great. So maybe it was just the share that ended up getting fouled up. Okay, so let's try the share again, and then you guys tell me if you see it. I think you have it. Do you guys have it? Okay,
Yes.
great. Okay, so we'll go from there, okay? So we're talking about saprobic fungi. Saprobic fungi are doing a lot of this breaking down, decomposing of dead organic matter. This is absolutely key for continued life on the planet because we have cycles for important elements, the carbon cycle, the water cycle, right? All these different things. And it is saprobic fungi that are kickstarting the recycling of all of these compounds, okay? And they do it in several different ways. I talk about carbon here. I talk about simple sugars being broken down from complex polysaccharides, okay? And nitrogen. This is another important thing, right? Nitrogen often gets locked up in things like proteins. Nitrogen is a necessary component for amino acids. Amines, right? Those of you taking orgo, you're probably overly familiar with this at this point, right? So amines means something related to nitrogen. And so all those amino acids have nitrogen in them, and this is one of the ways that that nitrogen is able to be released, okay? Okay, so now let's talk about spore production. And here you can see a fruiting body, a mushroom, releasing spores, and how this is a great shot, to see how many spores there can be and how sort of active spore dispersal can actually be, okay? These fungi are going to produce huge amounts of spores in those fruiting bodies, those specialized fruiting bodies that are related to reproduction, okay? This especially happens when nutrients are running low. I can't stay here indefinitely. It may be time to put resources into reproduction and send out spores so that my genetic lineage will go on, okay? Spores are very tiny. They're easily dispersed in the wind. You can see here, I mean, it looks like smoke, right? And it can easily be wind dispersed. And so we can do some simple back of the envelope kind of calculations to say how many spores there are, how many are likely to land on a favorable sort of location, right? And then be able to germinate from there. So now let's talk about parasitic fungi. Many species are parasitic, okay? You can have facultative parasites. That means that they grow on a living organism, but they can also grow on their own, okay? they can't only survive on a living host. And then there are obligate parasites. They definitely can only survive on a specific host. Okay. How does it work? Well, for many parasitic fungi, it works along with their hyphae, right? Hyphae, again, long, tubular, thin, they are very well suited to invading tissue. Particularly, there's lots of fungal parasites of plants. So they're very good at invading plant tissue. They enter the leaf through the stomata. Remember we talked about the stomata being on the underside of leaves. They open and close to allow gas exchange and water exchange. If a plant is wounded, it can enter through there. They're very good at penetrating those tough cell walls of plants. Some hyphae will produce this haustoria. These are projections that go into the cells. They don't break the cell membranes. They basically, it's almost like a fungal straw that gets put into a cell so that it could suck out, for lack of a better way of saying it, suck out some nutrients that are in that cell. Okay, but it's possible for these parasitic fungi to have symbiotic relationships where they don't kill their plants, but they're feeding off of the plant. It's interesting, excuse me, and it highlights perhaps some of the ways that these relationships, these symbiotic relationships, perhaps started. Okay, pathogenic fungi. Pathogenic fungi are a major health issue for humans as well as other species, okay? It's a major cause of death in people suffering from HIV and AIDS. There are plenty of other fungal pathogens that affect people that you are probably aware of. Things like ringworm, athlete's foot, jock itch, yeast infections, and something called tinea versicolor, which is a yeast that actually lives on your skin and will actually take nutrients right out of your skin and causes some discoloration. Fungi are a very large class of pathogens for plants, which means it's really important to understand these plant pathogens in terms of agriculture. They cause billions of dollars every year of loss to crops. Here is one, and it also has one of the best names of any fungus, corn smut. Okay, so here's a picture of corn smut growing on an ear of corn. This is a fungus that invades into the corn kernels, invades those cells and grows there. Obviously, corn smut ruins the ear of corn. It is no longer able to be used for the purposes which it was grown. Now, I will tell you that well, cotton candy, Joseph, that's interesting that you say that. I don't know if I would look at this thing and say, I wonder how it tastes. But yet, people do this a lot with fungi, right? They look at it and they say, I wonder if I can eat that. And corn smut in some parts of the world really is known as a delicacy, something that you can prepare in specific ways and then eat. So yeah, it's amazing when you look at fungi, some of the fungi that we eat as humans, and I always wonder about the first person who was like, yeah, I'm gonna eat that. I don't think I'd be that brave. Okay, let's talk about another important group of fungal pathogens that are attacking amphibian species, and this is called chytrid. Okay, chytrid is a group of different fungi, but they are all very similar in how they act. It's endemic to southern Africa, but it has spread around the world and is causing major problems for amphibians around the world. It basically infects the amphibians on and grows on their skin. Okay, and for an amphibian, when you have a severe chytrid infection, basically what you do is you grow a thicker skin. And it's effective in keeping the chytrid sort of from taking over the whole body. The problem with that is that amphibians breathe through their skin. So you make a thicker skin, it's harder for you to breathe. And so for many frog species in particular, they are suffocating. They're actually suffocating because their skin is so thick they can no longer efficiently take in oxygen. I've included a video at the end. I've included a few kind of interesting videos for you at the end. One on chytrid. There's a couple that I label as sort of sci-fi horror that you may want to check out for some specific fungi that infect insects, and it pretty gruesome. Well, how does it attach, Adam? That's a really interesting question. Remember those rhizoids, right? But also think about like that tinea versicolor that can infect a human. It just grows on your skin, and this is unicellular, and so yeah, a spore will land on it, right? And then the spore can germinate, and right, and it's taking the resources it needs, that absorptive heterotrophy, right off of that amphibian skin. Okay, this is causing a major, major problem for amphibian species, particularly in South America, in the tropics, where there are many species of tree frogs, really small frogs, often very colorful, and they are getting infected, and it is killing them, and they're declining very rapidly. Okay, now we go from one extreme to the other. Fungi are often mutualists, right? They can develop these symbiotic relationships where they live with a partner species, oftentimes in permanent contact, and that relationship can be beneficial to both participants. One great example of this are lichens. Lichens are a fungus and a photosynthetic microorganism that live together. There's 30,000 species of lichens, and they live all over the earth. I mean, they have one of the widest ranges of a group of species anywhere, right? They live in incredibly harsh environments. There are lichens in Antarctica. There are lichens above the Arctic Circle. This is an incredibly important food source for other mammals that live there. So, for instance, caribou, right? Reindeer that live really far north. This is a major source. Instead of, they can't always eat grass certain parts of the year, but they can eat this lichen. Okay, most of the time the fungus is partnered up. Well, wait, let me try and tackle this the way that I put it together, okay? So the fungal partners are what are called sac fungi. They have this little sac, this ascomyota, sort of the makeup of the fungal cell itself, okay? Some of them can grow on their own without their symbiont, but not really. You know, most of the time they really need their photosynthetic partner. So the symbiotic relationship is going to have usually a species of algae that is capable of undergoing photosynthesis, so making food from sunlight. Okay, and this relationship allows lichens to grow on exposed surfaces, right? Because you can if you have a photosynthetic partner that's going to create a food source for you, you can attach to something that life can't usually attach to, like a rock. And you'll often see lichens growing on moist rocks, tree bark, things like that. Okay, and again, in a place like, you know, near the poles where there's not a lot of rich organic soil to grow in, this is what you're going to find. Okay, I mentioned reindeer moss, and it can take different forms, right? This crustose form, this foliose, and this fructicose form, and I included a nice photo from your textbook here, where you can see some of the examples of those three forms. Okay, when you zoom in, this is sort of what the anatomy looks like. You have these hyphae, and it's thicker on the boundaries on the top and on the bottom. The bottom, you'll get those rhizoids that help anchor the fungus to things like this rock. And in the middle, you'll have sort of more space, some airiness, you know, in the hyphae, and that's where the algal cells are going to grow. The hyphae take up the nutrients that they need to survive from the photosynthetic algae, and they are providing moisture from the environment, protection from the environment. The algae are not going to dry out. Importantly, the fungi get fixed carbon from the photosynthetic cells. That's their carbon source. That's their food source, okay, their source of energy. Okay, so let's do an eye clicker question. All right, and this is an eye clicker question that I think is a good one. It helps kind of, you know, ask you to put things together across numerous lectures. Which of these is the best hypothesis as to why lichens have maintained such a strong symbiotic relationship with algae for so long? It's about another 15 seconds or so. Okay, couple seconds. All right, let's stop the poll. And let's see. All right, so about half of you chose C. Having a symbiotic relationship when colonizing harsh environments is beneficial. That is the best answer. Okay, C. Think about it. Anytime you're going into a situation where you can have some harsh environmental conditions, having a partner is always going to be beneficial, right? Where a partner where the relationship is mutualistic. You're giving something, but you're also getting a benefit, right? So that is true, and it's also true not just for something like lichens. It's true for many different species. One of the things that we notice in biology is that cooperation between individuals and cooperation between species often increases when environmental conditions degrade. Okay, so just it's not a law or anything like that, but it is a good general rule that you can use to sort of interpret the natural world. Okay, so why is A wrong? The algae fixes nitrogen for the fungus symbiont. What? No, bacteria fix nitrogen, not algae, right? Having a symbiotic relationship where one is photosynthetic is beneficial at high latitudes. That's actually not correct, right? Because at high latitudes, certain times of the year, the amount of sunlight that reaches really goes down. So being photosynthetic gets tougher as you move closer to the poles, as you move to higher and higher latitudes away from the equator, not better. We talked about C, and then D, reproduction is easier for the fungus, or reproduction is easier for the algae. Remember, they're tied together, so how they reproduce is really also tied together, okay? So now you guys had some comments. Is it Okay. Is it beneficial to the soil due to the fact that they grow on rocks? Well, Adam, that's actually a really good question because lichens, remember we talked about this before, right? This idea that when we talked about plants, that plants help create more soil by breaking down things like rock and things like that. Certainly the lichens attaching to rocks, they're going to speed up the breakdown process and increase the generation of soil as well. Okay, let's see. Did I lose the share? Oh, I think I lost it. Yeah, I lost it. I got to start it again. Sorry, guys. I do lose it when I try and click something else sometimes. It's so weird. Anyway. Okay, I think I got it back. Here we go. All right, great. So let's talk about fungi mutualists, right? And we're going to talk about several different groups. One that we're going to talk about are the mycorrhizae, okay? Mycorrhizae are these associations at the roots of plants between a fungus and a plant. Really, really important for plants, okay? Many vascular plants, if not most vascular plants, depend upon these associations. It's critical for the plants if they want to go into different terrestrial environments to have this mutualistic partner. And it happens at really small scales, just a lot of it, right? Plant root hairs often aren't able to take up enough water. And remember, fungi, because of those hyphae, are really good at taking up water. Very high surface area to volume ratios, okay? So they can take up that water, take up those minerals. You have ectomycorrhizae. These are fungi that wrap around I was about to say tree, but the plant roots. Doesn't have to be a tree, right? Any plant. Plant roots, but it doesn't penetrate the cell walls, right? It's just wrapping around. And this provides sort of a web around those roots that help. One way to think of it is increasing the surface area that they have to take water up, okay? And then you have arbuscular. How's that for vocabulary today? Arbuscular mycorrhizae. The hyphae do enter the root. They do penetrate those cell walls, but not into the cell membrane, just to the cell walls, okay? And this is how they help with nutrient, water, mineral uptake, okay? Mycorrhizae are essential for many vascular plants. Not only do they help with water and mineral uptake, but what's in it for the fungus? The fungus is going to get access to sugars, to amino acids, things like that. The fungus, interestingly, can also provide hormones. The fungus produces chemicals, biochemicals, that will impact the plant, okay? And it's interesting, fungi can do this for not just plant species, but some animal species, and that leads to those sci-fi horror videos that I have for you at the end, okay? Trees without ectomycorrhizae tend to not grow well. So when we talk about having healthy forests or conservation biology, where we're talking about reforesting and things like that, it's not just about planting the tree. It's also about getting the right fungus mutualists into the soil along with those trees, okay? Lots of crops, we have learned, need to be inoculated with the correct fungal mutualists in order for the plant to grow well, particularly if we're trying to grow a crop in a new environment, a different part of the world. How does this all work? So, for instance, certain crops, right? Think cranberries, which cranberries come from the North American continent, sort of the northern half of the United States, southern parts of Canada. That's the native areas for cranberries. They live in boggy environments, really nitrogen poor. Okay, they really need those mycorrhizae. Remember how fungus is good at breaking down proteins and releasing those amines. This is really important for releasing those amines and getting the nitrogen out of there. This is really important for plants. Nitrogen is a very important element for plant growth. Plants without chlorophyll also always have mycorrhizae, which they will share with photosynthetic plants. Okay, endophytic fungi. These are fungi that live in above ground parts of the plant, not just underground. We can also have above ground, okay? They produce alkaloids that can give a plant resistance to pathogens. So how do they have this beneficial relationship with plants? They can help protect the plant from other pathogens, particularly things like insects, insect pests, and bacteria. Okay, but oftentimes the relationship really isn't clear, and that's what that last bullet point basically means. Okay, okay, types of asexual reproduction in fungi. Okay, we already talked about spore production, so you can just produce haploid spores in the sporangia. You can produce haploid spores. Some fungi produce haploid spores at the tips of their hyphae, this conidia. You can just have budding, right? We talked about budding, which is a form of binary fission. I talked about that when I showed you the photo of the yeast up at the beginning. And then you can have breakage of the mycelium. Remember, the mycelium is the body, this bundle of hyphae. Okay, all right, now let's stop for questions. Somebody just had a question that they popped up in the chat. Did they want to just ask it? Okay, they don't want to just ask it. Rodney, go ahead. Rodney, you have your hand up. You want to ask your question? Oh, yeah. Okay, it's okay. Audio problems. All right, let's see if we can try and address the questions in the chat. Do carnivorous plants that need to manually obtain nitrogen from eating things also rely on mycorrhizae? Well, think about where those nitrogen needing plants are, right? So things like the Venus flytrap or pitcher plants, things like that. They're living in sort of boggy kind of conditions, which, by the way, boggy conditions is also the kind of environment where you would grow something like cranberries. Okay, so nitrogen poor. And do they have mycorrhizae? It really depends on the species, whether or not they have these relationships, to the best of my knowledge. But I am neither a botanist nor am I a mycologist. So, but that is a really super interesting question. Okay, would mycorrhizae that is invasive to a region be parasitic, or all mycorrhizae beneficial to the plant's growth? Yeah, that's another good question that I can't answer 100%. But mycorrhizae that have these beneficial relationships, right? If you have an established beneficial relationship, it really makes sense to stick with that beneficial relationship. Breaking that beneficial relationship could be disastrous for both of those participants. You want an example, right? Remember when we talked about corals and the symbiotic relationship? They have a symbiotic relationship very similar to the one that lichens have, where you have a photosynthetic partner. Right, and then what happens is that corals, when they're stressed, they'll actually eject their photosynthetic partner symbiont, and then both end up dying. right? So, so when you have beneficial relationships, it really makes sense to just stick with it. Now, speaking of sticking with it, did I lose the share? I think I lost it again. Okay, so we will stop the share and try it again. Okay. Got it? All right, great. Okay, so let's talk about how we classify taxa of fungi. There's 5 major taxa, okay? You can see them here. I already mentioned the chytrids. You have the microsporidia, the zygospore, the arbuscular mycorrhizal fungi, I mentioned those, and the dicaria, okay? Now, DNA analysis has shown that the reduced parasitic microsporidia are related to the fungi, and that chytrids and zygospore fungi may not be monophyletic groups. What the heck does that all mean? Okay, this microsporidia, very closely related to the fungi, but there is some debate about whether or not they are true fungi. Okay, the chytrids and the zygospore fungi, which you see here, the reason that these lines are dashed is because they are not monophyletic. They don't go back to a single common ancestor easily, okay? So they may have picked up many genes from other branches along the way, okay? So just something to be aware of. All right, sex in fungi. Sex in fungi gets very complicated very quickly. Okay, so we're going to try and break it down, but this is a complicated subject. Okay, sex in fungi, there are not sexes in fungi. There's no male, there's no female. There is what are called mating types. There can be many mating types, and you mate with a different mating type, okay? Mating actually happens when bodies fuse. So for instance, this happens a lot in mushrooms, and then they exchange nuclei. Okay, now they can remain fused for a short period of time, they can remain fused for a long period of time. Okay, so again, gets complicated very quickly. Those microsporidia, these are that I mentioned before, these are unicellular, they're typically intracellular parasites of animals. There's about 1,500 species, lots of chitin in their cell walls. They don't have mitochondria. They have reduced structures called mitosomes. So mitochondria that helps produce the energy, the ATP for cells. They don't have true mitochondria. This may explain why they are obligate intracellular parasites. They need to be close to an energy source that they need. They typically infect insects and aquatic species like fishes and crustaceans. Okay. That's okay. All right. Chytrids. I mentioned some chytrids before, infecting the amphibians. One of the ways that they infect amphibians is because they survive in water, and so the amphibians, of course, will often be in water. They are typically unicellular. They will have these rhizoids that anchor them to their hosts. Okay. What did I want to say with this slide? I don't even really remember why I wanted specifically to have this slide. Most of the other groups of fungi are terrestrial. Gametes aren't motive, so liquid water is not required for fertilization. The cytoplasm of individuals. I don't remember what point I wanted to make with this. This is what happens when you, I guess, start getting old. Okay. Zygomycota. Sexual reproduction here has to do with hyphae, and again, have to be different mating types. They release chemical signals that basically cause them to grow toward each other. Okay, the hyphae then basically bind and they fuse, and then you get nuclei fusion to form what's called a zygospore. This is the only diploid cell in the entire life cycle of zygomycota. Okay, otherwise they are haploid. Okay, in favorable conditions, the zygospore will actually undergo meiosis to create these sporangiophores they'll then contain haploid nuclei that go into spores. Okay. The thing is, zygospores are common. They're really everywhere. They are saprobes, they're parasites, particularly of insects and other invertebrates. Okay, there's well over 1,000 species. Many bread molds are zygospores. They really everywhere. Okay, arbuscular mycorrhizal fungi. I mentioned these before. These are symbiotic, right? Again, the plant roots. Look at what percentage of plants actually have these. Okay, 80 to 90%. It's amazing. So plants on land really depend upon fungi for a lot. Okay, that was the major thing that I wanted to do. The dicaria. Dicaria include the sac fungi and the club fungi. Okay, now here's what's really interesting about the dicaria. Dicaria are neither haploid nor are they diploid. The best way to describe them is not N or 2N, but actually N plus N. Okay. This is really interesting. The hyphae, this is where they get their name, the dikaryotic, right, where it's N plus N. The hyphae, the genomes themselves, right, actually they're both present, but they stay separate. Okay, which is just really interesting. And again, highlights one of the things that I told you very early on in my time with you. In biology, if it can happen, given enough time, it eventually will happen. Fungi are the proof of that rule. Okay. Okay. So Ascomyota. Ascomyota are the sac fungi. They are typically associated with water. They need a lot of water in order to survive. Many of these are the lichen sort of partner up with lichen. They produce these sacs called asci, right, which contain the ascospores. The asci then get incorporated into these fruiting structures that are called ascoma, and then that's how they release their spores. Okay, the Basidiomycota. I specifically wanted to mention the Basidiomycota. If you have spent any time in forests of any northern half of the United States, you have seen these, right? These are club fungi. You'll see them as mushrooms. You'll see them as those shelf mushrooms on trees. Amazing colors. Right here, I will never forget one time I was in the forest that I saw this Basidiomycota. Such an amazingly bright, vibrant, orangey color. You could spot it from so far away in the woods. It almost looks like it's glowing. It's so bright, in the subdued sort of light in the forest. And it's amazing how easily you can see. Many of these are edible. People who go mushroom hunting, right, in the woods, these are many of the species that they are after, that humans actually prepare as delicacies. Okay, how we doing? We're starting to run out of time. We're okay. We've got a few more slides. Okay. Sexual reproduction in these Basidiomycota. The part that you see, remember that mushroom is the fruiting body. Okay. The basidia, the spores actually grow on specialized structures called gills. For instance, on what we typically think of with a mushroom, let's say a button mushroom. If you look and if you can buy button mushrooms now in supermarkets, so the next time you have a chance, turn the mushroom over, you will see the gills, these specialized cells that will hold the spores on the underside. Okay, and they're called gills because they kind of look like gills. They look like paper. You know, if you look at a book, at its edge, all the edges of the paper, it kind of looks like that, sort of zoomed in. Okay, so the last content item, key concept that I wanted to mention was how fungi have practical uses for humans. And I've already mentioned some of them, right? Brewing and baking and alcohol production. But there's lots of others, right? We've already mentioned food. You know, but even beyond that, there's even more, right? So for instance, it's a brown mold that actually is used to create soy sauce, right? So, and this is a major product that many people depend upon for nutrition in many parts of the world. Other species from this genus, Aspergillus, can grow, which is a kind of mold, brown mold. It can grow on grains, it can grow on nuts, and it actually produces. I'm sorry, somebody's mic is on. Sorry, did somebody have a question? Oh, okay, sorry guys. And they produce carcinogenic aflatoxins. These are things that can actually be used in very specific things, including non-traditional medicine. Penicillin, which is the first true antibiotic, came from a mold, a green mold. Many different antibiotics have been discovered in molds. There are, you know, other things that have been used, other fungi that have been used as medicines for very long periods of time around the world. We use fungi for specific types of cheese making, right? So if you've ever had any sort of blue cheese, gorgonzola, those kinds of things, that is a mold that's growing in the cheese that is specifically introduced into the cheese in order to be able to grow, and that gives those flavors, those desirable flavors. Okay, lichens are actually used in parts of the world as a very inexpensive, easy to keep track of, easy to grow indicator of air pollution, because lichens are actually very sensitive to changes in air quality. Okay, fungi are important to the carbon cycle, right? They break down I already mentioned, I think, hopefully stressed, the role that they play in recycling chemicals and compounds. Fungi are also used in places where there have been toxic spills, oil spills, things like that, because the fungi can break down the things that have been spilled into the environment, and then they can be naturally recycled or more easily cleaned up. So, and then I've got the additional resources that I mentioned for you guys. The fungal video up at the top, it's a little bit long, but it's actually quite informative. I've got a video specifically on chytrid and some of those for lack of a better way of saying it, zombie creating fungi. So you may want to check those out. Okay. All right. So let's stop the share. Okay. And let's see if you have any questions in the last few minutes we have together.
Hello, Professor.
Hi.
Obviously, there's a lot of material on these slides. Like, what would you recommend? Obviously, your opinion, like, is the best way to like process all of it, maybe study or.
Yeah. So this is a kind of question that I've gotten in many times and in many different forms from you guys over our time together. Okay. And I have stressed, and I'm going to stress again, go back to the beginning. with the key concepts. If you look at my slides, if you look at the things that I stress, I take key concepts which are also highlighted in your textbook, and many of you feel like, well, it's just the textbook then. It's not. I want you to focus in on what the key concepts are. They will divide up the material from every lecture, from every chapter, and they will tell you, this is, think about it in terms of the key concept. Do I understand what this key concept says? Do I understand what it entails? Where do I need to sort of put the effort in? You may understand it to, sort of a first level. And then, okay, fine, I understand it to a first level. Go to the next one, go to the next one, go to the next one. Here we had 4. Now you check all 4 from this week's material, and then you say, okay, I understand them all at the most sort of base level, the most fundamental level. Then go back and say, do I understand it at a deeper level? Okay, and I think that if you do that, you will have a good sense of where you are in understanding the material and where you should spend your time studying. And you have different resources. So start with your notes, start with the slides. If you don't understand what's on a slide, then you go to your next level of resources. You can come and speak with me. You can go into your textbook. Each one of your chapters has that visual summary, which also includes questions. You could check that out as well. Is that helpful?
Yeah, it's very helpful. Thank you. I didn't think of that.
Okay. Yep.
All right, have a good one, Professor. Thanks for everything.
You too. And we've got a minute left. So guys, if you need to go, please feel free. Thank you all very much for spending this time with me. I enjoyed it. I hope that I was able to help you increase your understanding of this material. And Dr. Arnott will take over on Tuesday. But I'm not leaving just yet. So if you have questions, please hang around. And I'll try and do the best I can in answering them. Anybody else have questions? Rodney, you have your hand up. How are those audio problems?
Yeah, I'm back. I'm great. Yeah, so I got an email. You answered a lot of my questions in the previous response that you just to one of the other students. But I wanted to know when might be the next time you have some office hours, because I just wanted to talk about better test taking strategies and some other things.
Yeah. Rodney, shoot me an email and, let me know when you're available for an appointment. So, and we'll try and find a time. So if you said best day for me is, our Wednesday afternoons, like we'll try and find a time next Wednesday, that kind of a thing. Rather than having dedicated office hours, because so many students have said, because there's so many of you have said, oh, I can't make your office hours. I thought it would just make more sense to try and respond to individual requests for appointments. So just shoot me an email and request an appointment.
Okay, great. Thank you so much, Professor.
Yeah. Anyone else?
Professor?
Yes.
Hi.
Hello.
Hi. I thought I'd put my camera on and just thank you for a wonderful half of the semester.
Oh, well, thank you very much. I appreciate you turning the camera on. I never wanted to ask, but sometimes it's hard to lecture to a blank screen.
I can imagine. Thank you so much for everything. I really enjoyed your teaching style.
Thank you. Thank you for listening. Did you have a question, or was it just a thank you?
No, just wanted to say thank you. If I have a question, I'll shoot
thank you very much. I really appreciate that.
Of course.
Thank you. Yeah.
Stay safe, okay, Professor?
Yes, you too. Everybody stay well. We're close to the end. We're close to the end here. Everybody, really, please continue following the guidelines. Masks, super important. And especially as it gets warmer outside, so much safer than indoor spaces. So utilize the outdoors as well.
Okay, thank you.
Yeah, of course. Thank you. Melissa, you have a hand up.
Yeah, sorry my camera's not on, but
No, no. Please don't misinterpret. I never wanted to ask somebody to turn their cameras on. I understand. We're all at home. Who knows what's going on at home, right? Like, I'm always worried about somebody coming in here and interrupting, or the phone ringing, or something like that. So, no, please don't worry about it at all. But please, what's your question?
So my question is about iClicker. It's just like a logistic thing. So while I answered it, I realized that my answer was wrong and I tried switching my answer. Do you know how long in between like you ending the question or something like that, like how much of an interval I have to change my answer? Because I know we can,
No,
but I don't know like how long of a time we have to do it.
Right. No, I don't know. And I think that's probably pretty variable giving internet connections and everything else. But again, I don't so much want everyone to worry about, the getting the answer correct. If you got it correct, that's fantastic. If you didn't, I want you to learn from the question. That's what's most important to me. Overall, please bear in mind that the iClickers are worth very few points, right? Like, I mean, they're worth a point in essence, and so it's yeah, you lose the point, but it's not a huge impact on your grade, right? So I don't want people stressing out too much about iClickers and whether or not they got it right. It's not like it's not like an exam question is my point. So I hope I answered the question, and I hope I sort of was able to give you a little sort of relief there.
Yeah, I mean, I guess I just got like a little nervous and then just chose the wrong one initially and tried changing it, but it didn't work. So
It's okay. Yeah. No, don't worry about it. I mean, today I asked one question, right? So you get a point just for using iClicker, and then, let's say you got it wrong, right? So you lost one out of two possible points, and it's not the same 2 points as value once we weight everything, right? That an exam question that was worth 2 points is actually worth in terms of the course, right? It's worth much, much less than the exam question. So just keep that in mind.
okay, thank you.
You got it. All right, there's still about a dozen of us, so if you have questions, ask them. If we don't have questions that we want contained in the recording, I can stop the recording and then we can sort of tackle those. Okay, so I'm going to stop the recording.